Hey everybody. I recently did a video which I called How to Make Your Own Eucharistic Miracles at Home, and that video primarily focused on two papers. One was called The Relics of Jesus and Eucharistic Miracles, Scientific Analysis of Shared AB Blood Type. That one was published in Forensic Science, Medicine, and Pathology on October 30th, 2024. And then just a month later, in November 2024, the second paper was published. That one was titled Scientific Analyses of Eucharistic Miracles, Importance of Standardization in Evaluation. And that one was published in the Journal of Forensic Science and Research. Both of those papers were authored by Dr. Kelly Kearse. Last week, I had the honor of speaking with Dr. Kelly Kearse on my friend Dale's channel from the Real Seekers channel. In today's video, I am going to play some of my favorite highlights from Dr. Kearse's lecture on Dale's channel. And then I'm going to discuss a brand new Eucharistic miracle very briefly. But if you want to watch the full interview on Dale's Real Seekers channel with Dr. Kelly Kearse, in which I was there and I got to ask some questions, please see the link in the description down below. All right. I will be playing two clips from this nearly 2 hour long lecture on Dale's channel. Again, for the whole lecture, please see the link in the description down below. The first clip comes from about 40 minutes into the lecture. Dr. Kearse is explaining why he thinks that the data from Lanciano is not as impressive as some people think. Some people here includes Dr. Serafini, the author of A Cardiologist Examines Jesus, who wrote regarding Lanciano that the, quote, flesh and blood belong to the human species, end quote. That no uncertain terms there. Dr. Kearse's Dr. Kearse's point was that cross-reactivity is a big concern, and cross-reactivity was not discussed at all in Dr. Lannoli's report. Let's hear what Dr. Kearse has to say.
I would also point out that cross-reactivity from an immunology standpoint is a major concern. So the question I would have, well, does this antibody preparation exclusively recognize human blood? That's not demonstrated in the study, even if all of those other tubes were included. So let me just talk a minute about how this kind of thing often happens. This is the same kind of thing with the shroud. Well, they say they used anti-human antibodies. Why are you saying that it doesn't really show that it's human? So when you get a vial of something that's an anti-human antibody, so it's you buy it because, well, it reacts with human products. All that anti-human designation refers to is what the antibody was made against. So when it says anti-human, what that means, it means that a human protein was used to generate that antibody. So usually put this in another species and it makes antibodies. So it's anti-human is how it was made. It was made against a human protein, but that doesn't mean that it only reacts with a human proteins. It can react with that same protein in a lot of different species. So this is kind of a misunderstanding. I think a lot of even investigators, scientific investigators, they don't take it past the label and really look into this further. Now, this is a modern day screen capture from an antibody catalog, just a random antibodies, just to kind of show you how this works. So over here on the right, they say known species cross-reactivity. So notice how they word it. They say it's what's known, which means there could be some others that they haven't really tested. But if you look at just one kind of picked out here, you can see this antibody, it reacts against rat, human, mouse, hamster, and chicken molecules. And some of these others you can see up here, they react with numerous types of species. So when you see this anti-human, it only indicates what the antibody was made against, not what it has potential to react with. So these antibody preparations used in this study, so you're talking back in the 70s, these are going to be very crude antibody preparations. It's almost a guarantee that these would react with blood from other species. So as far as claiming that it's demonstrated that it's human blood, I don't think you can make that claim, especially unquestionably demonstrated. I don't think you can make that claim, even if you're willing to include all of those other tubes, which I just.
So there you have it. I think that the main thing there is that the antibodies in the 70s were not of the quality that they are today, and even the antibodies of today all have cross reactivity. Cross reactivity was not considered in the Lanciano study from the 70s, and so the confidence with which certain people talk about the work done at Lanciano is unearned. That was my first key takeaway from Dr. Kiers's lecture. I'm only going to play one other clip. The second part that I would like to play is about how frustrating it is that Dr. Castagnon Gomez treats the lack of replicable DNA to be a confirmation of the miraculous nature of the DNA, right? So, no evidence means that it's supernatural, right? So instead of we can't find human DNA, so there's probably no human DNA there. No. That's proof that it's miraculous DNA. That should sound nutty to you because it is nutty. And Dr. Kierce lays out why it's nutty, starting at about the 1 hour, 13 and a half minute mark right here.
To analyze this, you need to see something more than that. So they say it's proof of the human nature of the tissue beyond doubt. But so let me just kind of point out with all of this, I've never seen just kind of a simple fundamental question addressed to show that a DNA signal is present that originates from blood. So it's a very straightforward thing to do. I've never seen it. I've seen, these kind of things with these other variable, like STRs and things like that. But I've never seen this kind of fundamental question. Well, how do you know that the DNA is coming from blood and not just skin? So let me just pop into that for a second. I've written about this in terms of the shroud, but the miracles, I think it really applies because the shroud may be a little tougher to get DNA in good shape from that. But from the miracles, I think you'd have a better chance. So here we have white blood cells, a particular type called lymphocytes. They have these immune receptors on their surface. Skin cells don't. But if you look at the DNA level, skin cells do have the DNA for these immune receptor genes. They just don't use that DNA. And what makes it different and that gives you the power of discrimination in lymphocytes as part of their normal development, they will rearrange and splice this DNA. They're the only cell types in the body that do this. They rearrange it and splice it so it's a much different version than what you find in skin cells. So this is just a lot more detailed slide that it only makes the point that these gene segments in skin cells, they're there, but they're separated by very large distances. Where in lymphocytes, they rearrange and splice these together so it looks much different. And it's a very straightforward kind of analysis that you can do. And you have two different types of immune receptors you can look at and several different gene segments within those receptors that you could look at. So you have a lot of potential shots here to examine that kind of thing. And again, I've never seen this even mentioned or suggested. A very straightforward demonstration that any kind of DNA study you do, you're always going to have potential for contamination with that. But this type of thing, the rearrangement, it doesn't happen in skin cells. So it's a very definitive type test to show that, well, this signal truly is coming from blood cells. I think that's something that should certainly be considered, but I've never seen it really suggested as far as the DNA part goes.
I think that's really key is that the blood DNA is different than skin DNA, and there are simple tests that can be done to differentiate the two, and those tests either haven't been performed, or if they were performed, they were not published. The results were never released. And that is a little bit shady. That should probably cause some skepticism in the person who is looking into these miracles. In Dr. Kelly Kiers's presentation, we talked primarily about the miracles that are covered in Dr. Serafini's book. Lanciano, Buenos Aires, Tixla, Sokolka, those miracles. However, there are a lot more Eucharistic miracles, and I don't think that everybody always knows about all of these different miracles. I don't want to call them commonplace, but they do kind of happen a lot, which is weird. I would like to share an article with you right here. Investigation underway for potential Eucharistic miracle in small Indiana town. This article was published on Thursday, February 27th, 2025. I'm recording this on March 2nd, 2025, just a couple days after this article was published. The Archdiocese of Indianapolis said that an investigation is underway into a potential Eucharistic miracle at St. Anthony Padua Catholic Church in Morris, a small town in Ripley County. The investigation follows reports of a possible transformation of the Eucharist, a sacred element of the Catholic Mass, into the physical body and blood of Christ. They interviewed somebody who says, we have such a small little town, you can drive through it and blink and you'd miss it. It means the world. It does. I think there's something special about our church here. Okay, great. Let's take a look at the picture that they provide. This is the picture of the miracle. If you ask me, that looks just like the mold that Dr. Kelly Kiers was able to grow in his unconsecrated hosts. I think that this is mold. The Archdiocese of Indianapolis said that a professional laboratory is assisting in the investigation. We don't know for sure yet, but if eventually that does come to be, that's great. That's something very special. That's a great sign that, yes, Christ's living presence is among us, Father Dumont said. So the jury is still out on this one as to whether or not this sample is mold or whether it's human flesh and blood. But I'm betting that it's mold. I'm also hoping that they follow Dr. Kelly Kiers's protocol here, or at least something similar to it, something better than what was followed in 20 years ago for Buenos Aires and for Tixla. But Kevin, why are you so confident that it's mold? Have any other Eucharistic miracles been proven to be mold? I mean, I guess not, because they're not miracles. But supposed miracles have been confirmed to be mold. And it seems like more and more recently, this is happening more often, that they are being confirmed to be mold. Here's an article on Sunday, June 19th, 2011, at St. Augustine Catholic Church in South St. Paul. A host was placed in water after it was accidentally dropped. It turned red. There's a picture of it. A fungus, not a miracle, caused the communion host to turn red this summer at a South St. Paul church, the Archdiocese of St. Paul in Minneapolis said Wednesday. The Archdiocese began an exhaustive biological analysis of the consecrated host after it was found on the floor of St. Augustine Church after Holy Communion. So it appeared to be the blood red of tissue. If I had not known what it was, I would have thought that there was maybe a small bloody piece of tissue. It was striking enough that there was no way I could have disposed of the remains of the host at that time with good conscience. So this mold, stuff that we have determined to be mold, is tricking people into thinking that it's flesh. Some people kind of laugh at the mold idea, but I don't know. I think that's quite likely. Additionally, here's a second article. Bleeding host was mold, not miracle, according to diocese. This is not the same one. So this one was from 2011 in St. Paul, Minnesota. This one is in Salt Lake City and 2015, not 2011. So in November 2015, it was alleged that a consecrated host from St. Francis Xavier Church in Kearns, Utah, appeared to be bleeding. A thorough investigation has concluded that the host did not bleed, but the change of appearance in the host was due to red bread mold. The consecrated host has been disposed of in a reverent manner, as is required. So, and look, word spread in the church filled with people. The host was on display for a short time. So they're so confident in this host that they're putting it on display before the results come in. And I think it's fooling people, because people seem to believe it until the genetic work is done and it's revealed that those are the genes of fungus, not of humans. That is why the work that Dr. Kelly Kearse is recommending is so critical. This needs to be done. I think that I think that Catholics need to be making this push. I don't know why it's me. I don't know why more Catholics aren't talking about this. Nobody's going to take me seriously on the Catholic side. I get that. That makes sense. But that means that it's up to you, Catholics. If you care about truth, and you're not willing just to kind of say, oh, maybe it's a miracle, and then leave it at, no. If you care about these things, then you should be pushing for scientific rigor. And with very few exceptions. Dr. Kelly Kearse, Dr. Stacy Trasankos, that's about it. I'm not seeing Catholics push for the kind of robust science that is necessary in order to confirm that these things are miracles. That is very troubling to me. And I would like to think it's troubling to Catholics as well, but I don't know that it is. Somebody left a comment under one of my videos recently asking me to talk to another Catholic content creator, one who's much bigger than I am, because he was talking about Eucharistic miracles in a recent video. I did DM this content creator and asked him if he's interested in talking, and he said no, which is fine. Not everybody needs to talk to me. But if you're not even willing to talk to me about it, you shouldn't be talking about Eucharistic miracles on your channel. And maybe they didn't know any better, and maybe they do know better now, and maybe they won't in the future. Who knows? But I think it's a very troubling sign that Catholics are so willing to believe these things while the robustness of the evidence is quite lacking. And I'm further worried by the lack of appetite for more robust testing. Everybody seems quite happy to accept the shoddy science performed by Ricardo Castellan Gomez's Grupo Internacional de Pax, or de la Paz, or whatever it's called. I think Pax is Latin for peace. And it's supposed. Anyway, that's all that I had for today's video. I just wanted to share Dr. Kearse's lecture, 2 of my key takeaways, and the news about the recent Eucharistic miracle. I think it's mold, but only time will tell. Thanks, everybody.